the skin cancer, but for the dark skin. We know that the people here in Indonesia don't like dark skin. Yeah, and then this is very interesting too because the synthesis is pink. Yeah. Yes, the pink. This is. We didn't need some coloring substance for the sun cream because the women want the skin like what's not pale, segar, like pink. This is beautiful woman, not pale skin, but with the pink, small pink like this.
Yes, really.
Okay, we have only 10 minutes for the discussion. Yes? Okay. 15 minutes. Okay. Thank you. We have any question? Okay, 1, 2. Maybe for the first session, please go to the mic.
Okay, thank you very much. Good morning, Professor. My name is Flandiana. I'm from Gajah Mada University. So it's very nice talk, and I do agree that the sun radiation is very dangerous for the skin. Like to mention, previously people are only talking about UVB, but recently research and publication about UV interested group is more talking about the UVA. And my question is, you show the result in vitro about the toxicity test, but have you ever tried to test how is the effect of the the potential agent to say like UVB or UVA induced DNA damage, or in vivo probably you can do like topically, you apply topically to the skin about murine skin or human probably with a very low dose and check about the sunburn cell, which is one of the indicator about the UV induced damage, because at the end chronically it may induce like skin cancer, but for acute we cannot get the skin cancer in a very soon after the irradiation, but long time we can get the skin cancer. For example, like when we perform experiment in the mouse, we need about 1 year to develop the skin cancer, but only one single shot we can already know about the damage or so on. So probably it will be good to test about the protective effect of the potential agent due to the UVB induced damage or protecting factor before we use it for the human, I think. And the second question is like, you show like some sun protecting factor have about 200 or 80 or so on, and probably not in Indonesia yet, but in other countries such as Singapore or some place in Europe also, they start to sell about the UV sunscreen with 100 sun protecting factor, 100 SPF. Meanwhile, there is a report saying that above the 50 SPF, there is not really so different result. So what is your comment about that? Thank you very much.
Okay, please. Sekalian dua-dua biar kalau.
Okay, thank you. My name is Nia. It was an interesting presentation. I have 3 questions for you. The first one, maybe I missed some information before, but would you mind to explain more? Why must be sufficient conjugated bond? Yeah, you have explained before. And the second, it's similar with a previous questioner. How to categorize which is SPF good or not? Because you said that 300 is good, 30 is good. I don't know it before. And the third one, what is your next planning about it? Because I'm sure that you want it to be applicable to our society. Thank you.
Okay, please, maybe. Yang SPF-nya tadi yang pertama yang mana? Can you?
Excuse me, with the second questioner, could you repeat the first question?
Chemistry.
It is about chemistry aspect in your explanation about sufficient conjugated bonds.
Oh.
It's not short or long, yeah, because I missed some part in that information. Thank you.
Yeah. Okay. Thank you very much. Good question, but not easy to answer. Yeah, I just the second question first. What we mean by sufficient pi bond conjugation? Yeah, in general, when we have about 2 or 3 aromatic ring, benzene group, joined by one carbonyl group, like oxybenzone, it is enough. Yeah. With cinnamon, it has only one aromatic ring, but conjugated with one double bond and one CO bond, and that's also act as a sunscreen. Yeah. Well, what we have is a bit more than that. We have one aromatic ring joined with CO and one aromatic ring, but it is it is multiply. We have 4 systems like that. Yeah, and I think that is the reason why our compound is it seems to be quite good. Yeah, to be quite good. It is different from when we have a straight very long conjugated by bond, like for example a beta carotene, for example, beta carotene. It is quite long. I think about 10 double bond joined with one benzene at one end, and the second is also at the other end is one benzene. With this sort of molecule, for example, beta carotene, it absorbs not in the UV region but in the visible region, about 500 and something like that. So it is not UV active anymore. Yeah. So in general, having 2 or 3 benzene ring joined by one CO bond is enough to have a compound to be active as a scan grid. Okay. And how to categorize that certain SPF is good and no good? Yeah. As a general rule, when we have SPF greater than 10, people say that is good as a scan grid. Greater than 10, that is good. Yeah. Of course, when we have much greater, that will be better. Yeah. So it is easy to measure SPF. Is only you run the UV spectrum in certain concentration. We have the standard